Hey there, my name is Nathan Lapierre. I'm a PhD student in computer science at UCLA, and today I'm going to be talking to you about our method Metalign, which is for efficient alignment-based metagenomic profiling using containment min-hash. So metagenomic profiling, just to define this term, a couple of words here. Metagenomics is defined as whole genome shotgun sequencing of microbes, such as bacteria and viruses, recovered directly from their host environment. Whether soil, ocean water, human gut, microbes are everywhere. And the advantage of this, obviously, over traditional culture-based analysis of microbes is that in culture, you can't always sequence all the microbes, and you can't capture the real community diversity in the real world. So that's metagenomics. Profiling is predicting the organisms present in the sample and their relative abundances. So outputting something like E. coli is 30% of the sample, S. aureus is 12% of the sample, and so on. And usually this is done with a reference database. So you have some reference genomes, known microbial genomes, and you have your sequencing reads. And the simplest thing you can think of doing is aligning these reads to the reference genomes. You can do something like BLAST. And the advantage of BLAST and other alignment-based approaches like Megan are that they're highly accurate, they're intuitive, even non-technical people get the concept, and alignments are useful. They can be used for all sorts of other analyses once they've been produced. However, the reason why people don't always do this is because it can be prohibitively slow. So most people feel BLAST is very accurate, but these reference databases can be hundreds of gigabytes in size, and you can have tens of millions of sequencing reads. So blasting all the reads against the whole database can take months. So a couple of other heuristics have been developed. Some k-mer matching approaches used by methods like Kraken-2 and its abundance estimation counterpart Bracken, as well as Clark. These methods tend to be very fast and have high recall, but they tend to generate a lot of low abundance false positives, which gives them low precision. And they also store these big k-mer data structures in memory, so they can have high memory usage. Marker genome-based approaches attempt to perform alignment, but only against a subset of genes called marker genes that help you differentiate between the different organisms. So these methods are often high precision and use low memory because they only have to store a reference of the marker genes. But some of the cons are low recall because you can't always align reads specifically to these marker genes, especially with low abundance organisms. And it's difficult to retrain a custom database, which some people are interested in doing. So examples are Metalign-2 and MOTUS-2. So I kind of mentioned that some of these approaches have high recall but low precision. Some have high precision, low recall. This kind of makes sense, right? It's easy to say, all of the organisms in our reference database are present in the sample. That would give you high recall, low precision. It's also easy to just confidently predict only a few organisms. That gives you high precision but low recall. So balancing precision and recall is kind of a key challenge. And alignment-based approaches can do that, but can we do that without the prohibitive runtime or memory usage? And our answer to that, hopefully, is yes, we can. Our method is called Metalign, and it's called efficient alignment-based profiling. And the intuition is that the main limitation to the alignment is the slow runtime, and that's primarily due to the massive reference database sizes. But a lot of these organisms, you would think they share almost no sequence with the sequencing reads, so you should be able to quickly rule them out without having to perform a full alignment. So if we can do that, then we can dramatically reduce the alignment time and make this analysis feasible. So that's basically what we do. In our method, first stage is this database pre-filtering step that I just discussed. Then we perform alignment on our filtered database. And then in stage 3, we process multi-aligned reads, which are reads aligned to multiple different genomes. And then we compute the output profile. Okay, so now I'm going to go through each of those steps. First step is pre-filtering with containment min-hash, which we call SMASH for short. SMASH uses k-mer sketches to estimate what's called the containment index, which you can think of as the percentage of k-mers in an organism that are also present in the reads. And it's been shown theoretically by our co-author, David Kozlicki, that for applications like metagenomic profiling, where there's a big set size imbalance, there's many more k-mers in the reads than there are in any individual genome. The Jacquard index is biased towards 0, so containment index is preferable. A well-known method is MASH, which uses the Jacquard index. So by doing this SMASH, we can use a really comprehensive reference database efficiently. In our case, we use all NCBI assemblies, so RefSeq, GenBank, even some assemblies that aren't on GenBank, to make a really comprehensive reference database. And we can use it efficiently by doing this pre-filtering step. And so the mathematical concept of containment min-hash was introduced, as I mentioned, by Kozlicki and Zabedi in 2019. But Metalign is kind of the first method to put it into practice anywhere, and in this case, into metagenomic profiling. So basically what we do is we take each genome from the reference database, we SMASH computes the containment index, and then we keep organisms with a containment index above a certain cutoff and retain them in our subset database. And in practice, doing so reduced our full database, which was 243 gigabytes compressed, to about half a gigabyte to 5 gigabytes, depending on the diversity of the sample in question. So this can really dramatically reduce the size of the subset database. And then we perform alignment from the reads against the subset database. We use Minimap2, which is a recent accurate and very fast aligner. So that we have our alignments. Then we want to produce this output profile. But to do that, we have to deal with multi-aligned reads. And these are, as I mentioned, reads aligned to multiple genomes. So they're ambiguous, but if we found that if you just throw them away, it hurts your abundance estimation performance substantially. So to deal with this, we apply a pretty simple two-stage approach. We find the relative organism abundances using the uniquely aligned reads only, and then we just save the multi-aligned reads in which organisms they align to. And then for the multi-aligned reads, we assign them proportionally to organisms according to their unique read abundance. So I have a little toy example here. So we have 3 genomes, 1, 2, and 3, that are 50, 20, and 30% of the sample based on the unique reads. And then read 3 here maps to genomes 2 and 3. And relative to this set of genome 2 and 3, genome 2 is 40% out of 20, it's 20 divided by 20 plus 30, and genome 3 is 30%. So we assign 40% of read 3 to genome 2 and 60% to genome 3. So we're not explicitly classifying reads, we're just trying to find the most accurate abundance estimation possible. And I kind of parenthetically mentioned here that we deal with all sorts of other funky scenarios where one paired end maps to one organism and the other maps to multiple, and there's a bunch of cases like that we deal with that I'll skip the technical details on. So that's basically the overview of the Metalign approach. We have the database pre-filtering step. We can dramatically reduce the database size, get the subset database, perform alignment with Minimap2. We deal with those multi-aligned reads and compute this output profile. Okay, so that's the Metalign approach, basically. So now let's just get into the results. I'm going to assume people are mostly familiar with these metrics, but precision basically measures false positive rate, recall measures false negative rate. F1 score is the harmonic mean of precision and recall. So the F1 score is high when the precision is high and the recall is high, and they're relatively close to each other. So it's measuring that precision recall balance issue. So that's presence absence prediction. Another question is how well do you estimate the abundances? For that, we use the L1 error, which is the sum of absolute value errors of estimated versus real organism abundances. So if we predict that an organism is 10% of the sample, but it's actually 60%, then we get an L1 error of 0.5. In the results I'm going to show, so this ranges from 0 to 2, where 0 is better. But in the results I show later, I've flipped it and rescaled it such that it's a 0 to 1 range where 1 is better, just to get it on the same scale as these other metrics. Okay, so those are the metrics. We looked at 2 different data settings. We wanted to look at two very different settings. So one of them is this contest critical assessment of metagenome interpretation. It's a high-profile benchmarking competition. It has 8 simulated data sets. They're high diversity. There's a lot of, I say missingness, meaning they have high abundance novel strains that nobody has in their reference database. So it's kind of really simulating challenges you face in real data. But of course, that can never be fully captured. So one thing you can do is also look at a mock community, and we use this one by Peabody et al. A mock community is basically a community of microorganisms that is actually created in the lab and then sequenced. So it's real data, but you also have the ground truth available. So that's why mock communities are really nice. And this one in particular is low diversity, and the organisms are very well characterized. There's no novel strains. So these are two very different settings. And so moving into the CAMI1 data sets, these are results averaged over all 8 data sets at the genus level. We compared against a lot of these other approaches I mentioned. What kind of stands out across all 4 metrics is that Metalign, MOTUS2, and Megan performed very well. As I mentioned before, some of these Kamer-based approaches like Bracken and Clark had low recall. I should mention here that organisms below 0.01% abundance have been removed from the predictions because Bracken and Clark tend to generate many of these very low abundance false positives. So to be fair, we cut those out, but they still had lower precision. And some other methods, for instance, the marker-based Metaflan2 had low recall and also poor performance on scaled 1 error because it's harder to estimate the abundances when you only know the reads that hit the marker genes as opposed to the whole genome. So MOTUS2 and Megan were also comparable in accuracy. However, when we look at the runtime and memory, Megan was actually a couple of orders of magnitude slower than the other methods tested, even using Diamond, which is a pretty efficient protein-based alignment method. So this is summed over all 8 data sets, which reach about 50 million paired end reads each. And most methods took around 10 hours. Kraken 2 was very fast. Megan was a couple of orders of magnitude slower, around 1,000 compute hours. Note that this is a logarithmic scale here. Memory, Clark used by far the most, over 150 gigs. Marker gene-based methods such as Metaflan and MOTUs had very low memory usage, around 2 gigs. And there's sort of this middle pack of using around 40 gigs of memory where Metalign fits into. So Metalign is middle of the pack in both of these. We're not trying to be the fastest method. We're trying to be the most accurate method that has reasonable speed. And looking at the other data set, the Peabody mock community, Peabody et al. So here we looked at the F1 score. The abundance estimation isn't shown because it's hard to know the ground truth for that. But we do know which organisms are in the sample. And we looked at various different abundance cutoff thresholds. So again, removing organisms from the predicted sample if they're below this abundance cutoff, which helps methods like Clark and Bracken. So when you do this, they generate a very low abundance false positives, but when you do this, it improves their performance. Metalign was the highest performing method or tied for highest on all cutoff settings. Both Metalign and Bracken had perfect presence absence prediction at 5% abundance cutoff, but you can see Bracken was much more sensitive to this threshold. MOTUS2, which did really well on the CAMI data, was actually generally the worst performing method on this data. And then finally, another question you could have is basically, is Metalign just doing well because it has a really comprehensive reference database? So I mentioned this issue of being able to retrain with custom reference databases. Kraken 2 makes this really easy. So we retained them on our very comprehensive reference database. Long story short, this did dramatically improve the precision and F1 score from the original database to the new one. However, it's still a little lower than Metalign in both of those metrics. And the really big thing, it's still very fast, but the really big thing is that the memory usage shot up to 325 gigabytes because it has to store a big Kamer data structure in memory. So this becomes memory unfeasible for most users. So to recap, Metalign was the only method that performed well across both data sets with reasonable runtime and memory usage. Non-alignment methods tended to struggle in one setting or the other, and most non-alignment methods had low precision or recall. Megan, the other alignment-based approach, was comparable in accuracy to Metalign, but the runtime became infeasible with large data set sizes. And retraining Kraken 2 on Metalign's comprehensive database improved the results, but the memory usage became infeasible. And so Metalign is able to achieve highly accurate results by leveraging an alignment-based approach in combination with a highly comprehensive reference database. That makes sense. The challenge is, how do you do that with a reasonable runtime and memory usage? And the way we're able to is using this smash-based pre-filtering stage. So the pre-filtering stage, as I mentioned, it dramatically reduces the database size, often by a couple of orders of magnitude, which makes the alignment runtime feasible. And I didn't go into this as much, but Smash uses these lightweight K-mer sketch data structures, so it's also memory efficient. So that's how we're able to get the accuracy of this alignment with reasonable runtime and memory. Okay, so that's the basic summary. I would like to thank my co-authors, David Kozlicki and Sergey Mangel, who co-supervised this project. Eliezer Eskins, my PhD advisor. I'd like to thank Mohammed, who helped out with a lot of the Kraken 2 experiments. And I'd like to thank Lana for her help on the manuscript, which is available on bioRxiv. This is a bit of an old version, but we're hoping to have it in journals soon. Our package is available on GitHub. Metalign is also available on Bioconda. So I encourage anyone who's interested to check out the paper and check out the software. Thank you so much for listening.